stars the special monthly presentations by McGill scientists to talk about their research for support staff. We are on the traditional territory of the Haudenosaunee and the Anishinaabe nations, and McGill honors, respects, and acknowledges these nations as the traditional custodians of the lands and the waters on where we work, where we meet. My hope for our exchange is exactly that we continue this learning, this exchange, no matter where we are, and that we keep our spirits up, that the land will restore, the land will give back. Our speaker today will be introduced by Nicole Robinson-Paul.
Hi, everyone. Dr. Daiva Nielsen is an assistant professor in the School of Human Nutrition at McGill University. Her research program evaluates gene-environment interactions on nutrition and health outcomes with a specific focus on neurobehavioral aspects of eating, including food reward and sensory perception. Her lab also engages in knowledge of translation activities to advance nutrition research and practice, such as through the application of machine learning to organize dietary intake data and the use of clinical genetic testing for personalized nutrition. I'm super excited to hear your talk. Dr. Nielsen, the floor is yours.
Thank you so much, Nicole, for the warm introduction. And thank you so much, Ingrid, for welcoming me to be a part of this series. I will hide the video of the speaker just so we can have the focus on the slides. And then when we have the question period at the end, I'll put the video back on. So thank you again for the welcome invitation to speak. I'm really excited to share my work here. I'm very passionate about what I do, and I really look forward to taking your questions once I've finished my presentation. So I'm going to start off, because I'm fairly new to McGill, I'm going into my third year of being an assistant professor. I'm going to start off with some of my trajectory and my training. So I will go through some of my work as a doctoral student and a postdoc, just so you can see the evolution of what it is that I'm doing, because I have been building the focus and the activities since I was a doctoral student. So I'll go over some of my previous work and then get into my current projects that I'm leading here at McGill and share some of the preliminary results of those current projects with you as well. And my work is really focused around eating behavior. I called my talk this Beyond Counting Calories title because it does directly relate to weight management. And we know that our body weights are really directly tied to what it is that we're consuming. But really the focus of my work is on those ingestive behaviors or the eating behaviors that people have. And I'm interested in a diverse suite of factors that relate to eating behaviors, including genetics, but also the environmental exposures that we see on a day-to-day basis. So I'm excited to share some of the diverse aspects that I'm bringing together in my research program. So I start my talks with a couple of cartoons generally, just to set the stage. When I was a doctoral student, one of the big questions in the field of nutrition and genetics was, well, if someone were to find out about their own genetic information, what kind of influence would that have on their behaviors? And there's a lot of interest in all sorts of behaviors, whether it's physical activity or adhering to various medical recommendations or medical screening practices. But as a nutritional scientist, I'm really interested in eating behavior and what it is that people are consuming. So there have been a couple of scenarios proposed in the field that if someone were to have a genetic test done and learn about their own genetic information, it could actually serve as a source of motivation to encourage that individual to eat more healthily. Let's say they find out that they have certain predispositions to diet-related diseases such as diabetes or heart disease. Well, if they knew that and they knew there were nutritional strategies to offset that risk, they may actually adopt some very healthful dietary behaviors. However, there was also a counter argument that perhaps this information would lead to more of a deterministic or fatalistic perception, that having these genes makes it sort of a destiny that these outcomes would arise at some point, and that individuals might feel somewhat hopeless and that it doesn't really matter what they do with their behaviors. In my case, it would be the diet, and that they may just adopt behaviors that are not so healthy. So the kind of two opposing viewpoints, that knowing someone's genetic information could have a positive or a negative effect. And so at the time, this hadn't been studied as it relates to nutrition. And I was one of the first researchers as a PhD student to look at this. So I'm going to go over the field of nutrigenomics because that's really the aspect of genetic testing that I have a lot of interest in. So I think we can all agree that nutrition is related to health, and it's a pretty strong relationship. But what's also true is that our genetic variants can influence the way that we respond to nutrients in the foods that we consume. Now in genetics, there's typically 3 versions of a gene that someone can possess. It's referred to as various genotype combinations. And depending on which genotype combination someone has, their response to nutrition from the foods they consume can either relate to health in a way that increases a particular health outcome risk, decreases the risk, or has an intermediate or possibly no effect. And really the tenet of nutrigenomics is that one size does not fit all in terms of nutrition recommendations, and that we can further fine-tune the nutrition advice that's given to individuals according to their genetic backgrounds. And so I'll give an example of this framework with coffee and cardiovascular disease. Now, this relationship has been quite inconsistent in the scientific literature, and a number of studies have reported beneficial effects of coffee consumption on heart disease outcomes, but also a number of studies have reported adverse relationships between coffee intake and cardiovascular disease. But what previous studies have neglected to consider is the role of genetic variation that is involved in caffeine metabolism. So when I refer to coffee, I'm really talking about caffeinated coffee, not the decaffeinated variety. And CYP1A2 is a gene that's involved in the metabolism or the clearance of caffeine from the bloodstream. Now, depending on the genotype combination that one may have, they can either be a slow or a fast metabolizer of caffeine. And the slow metabolizers, those that tend to have caffeine stay in the bloodstream for a longer period of time than others, have been reported to have an increased risk of cardiovascular related outcomes, whereas those who are fast metabolizers of caffeine actually have been observed to have a reduction in the risk. I'll go over some of the pharmacokinetics of this genotype. So these individuals who possess the slow versions of the genotypes, it's been shown in previous research that the inducibility of the CYP1A2 genotype is much lower. So this enzyme that CYP1A2 encodes is called an adaptive enzyme. So it's really produced by the body when there's a need for it. So if caffeine were to be detected in the system, then the CYP1A2 enzyme production would increase. So it's different from a constitutive enzyme, which is produced at all times in the body. So it's really produced when there's a need for it. Now, individuals who have the AC or CC version of the genotype do not have as high of an inducibility of that enzyme. It's not produced at as high a rate as individuals with the AA version of the gene. So they produce much more of the enzyme, and as a consequence of that, they are able to clear caffeine from the system at a faster rate. And so this inducibility of the enzyme is what's related to whether or not someone is a fast or slow metabolizer of caffeine. And so that's been known for quite some time, and it has been applied to nutrition research more in recent years. And so I'm presenting previous work. This isn't work that I conducted, but it's part of the evidence around this field of nutrigenomics, and really some of the stronger evidence showing that dietary recommendations can be modified at the level of someone's genetic information. So this evidence comes from case control work that was done in Costa Rica. This is the Costa Rica Heart Study that consisted of about 4,000 research participants, half of whom had suffered a myocardial infarction or a heart attack, a non-fatal version of a heart attack, and the other half who were control participants who had never suffered a heart attack. And so these participants had a wealth of information collected on their lifestyle, previous medical history, and also their genetic information was assessed for the CYP1A2 gene. Now, these investigators, when they looked at the relationship of coffee consumption and heart attack risk, without considering genetics, they were able to observe that consuming more than 4 cups of coffee a day was associated with a significantly increased odds of having reported suffering a non-fatal heart attack at some point. And this is actually the general recommendation for caffeine consumption, is to restrict the amount of caffeine that someone consumes to the equivalent of about 4 cups of coffee per day. That is the current general recommendation for caffeine intake. So this is just the evidence when genetic information was not considered in the analysis. Now, when genetics was applied to the analysis, there was a different pattern that emerged. So amongst the slow metabolizers, these are the bar graph portion on the right side of the graph, these individuals had a significantly increased odds of reporting having suffered a heart attack if they consumed more than 2 cups of coffee per day. So not even at that higher level of 4 cups or more, but it was actually at a lower dose of the equivalent of about 2 cups of coffee per day. And if they were a fast metabolizer, there were actually no statistically significant associations with heart attack odds. And so this is part of that basis now in the genetic framework, that whether or not coffee plays a role in heart disease outcomes appears to be different depending on different genetic backgrounds. And so those who have the fast metabolizing gene for CYP1A2 had no significant association with a heart attack outcome. Now, what was even more striking is when this analysis was restricted to only individuals who are under the age of 50, so the younger individuals in the cohort. And the reason the investigators did this is because the risk of having a heart attack increases with age. And so to kind of evaluate this relationship without that extra consideration of knowing that the odds of a heart attack would increase with increasing age, they looked at this subset of individuals who are under the age of 50. And there they saw even more pronounced associations between the genetic information and the outcome of non-fatal heart attack. So still amongst the slow metabolizers, they're seeing this increased odds once you have over the equivalent of 2 cups of coffee per day in terms of the caffeine dosage. A good rule of thumb is that about every cup of caffeinated coffee contains about 100 milligrams of caffeine. So this would be the equivalent of about 200 milligrams of caffeine per day, consuming about 2 cups of caffeinated coffee. And the odds ratios have really dramatically increased in this younger subset. So it's gone to about a 3 to 4-fold increased risk of potentially suffering a heart attack if you were someone who had a slow metabolizing version of the CYP1A2 gene and consumed more than 2 cups of coffee per day. Now, amongst the fast metabolizers, it was an opposite pattern of association. And so there was actually a reduction in the odds of having reported a heart attack if someone was consuming moderate amounts of caffeine. So between 1 and 3 cups per day, that's what these 2 bars represent. So the message here is that the relationship between caffeine consumption and heart attack odds seems to differ according to the genetic background of the individual that's being assessed. And so the fast metabolizers of caffeine could actually have a protective effect if they're of a younger age demographic and consume moderate amounts of coffee, whereas slow metabolizers do need to be mindful of how much caffeine they consume and potentially could benefit from limiting that to no more than 2 cups of coffee per day. And that's quite different from the general recommendation that is currently in place, which is to consume up to 4 cups of coffee per day. So by applying the genetic information to the analysis, there's more precision that's revealed in terms of looking at these associations. And so that's really the tenet of this field of nutrigenomics, is to try and understand how do these relationships between constituents in food and various health outcomes differ according to our genetic backgrounds. Now, when I was a doctoral student, I was really interested to understand whether this information could alter someone's behavior, and particularly their dietary behaviors. In nutrition, we know probably better than anyone else in a health-related field that it's very hard to change behaviors, particularly dietary behaviors. There's a number of complexities involved, including emotional states and memories associated with food, economic considerations in terms of what people can afford to purchase. But it was an interesting question to pursue understanding whether knowing this type of genetic information would actually impact somebody's dietary behavior. So I designed a randomized controlled trial, which is one of the highest levels of scientific evidence, to study if we could provide genetic information, what kind of impact would it have on the dietary behaviors of the study participants. So I included a number of genetic variants in my study report for the intervention group. And so one of the pieces of information that they received was on the CYP1A2 gene and its relationship with caffeine and cardiovascular disease outcomes. So I've gone over that literature, and I'll just briefly summarize the other 3 pieces of information that were in the study. We also provided a genetic report for vitamin C status. And so a gene, GSTM1, that's involved in vitamin C recycling, has been associated with an increased risk of vitamin C deficiency. So that's actually low levels of ascorbic acid in one's bloodstream if they don't consume enough vitamin C from the diet. We also provided a piece of information for sugar consumption. I'm very interested in this field as well. This is the genetics of taste. And as I go into my work here at McGill, you'll see I'm pushing this forward as well in what I do now. But there is some interesting evidence that these genes involved in taste perception also relate to food intake and food preferences. So this TAS1R2 gene is involved in sweet taste perception, and individuals with particular variants in TAS1R2 have been shown to have a higher consumption of sugars than individuals with a different type of genetic mutation in that gene. And finally, we had a piece of information about sodium. The ACE gene has been implicated in salt-sensitive hypertension. So this is a form of blood pressure that is directly linked to one's response to salt in the diet. And individuals with a particular mutation in the ACE gene are at a higher risk of having salt-sensitive hypertension. So we provided a number of reports related to these genes and what the link is to diet and health. And we provided customized dietary recommendations according to the participants' genetic results to see if they would adhere to those recommendations. Now, it is a randomized controlled trial, so that meant in addition to the intervention group, we had a control group, which was a group of participants who did not receive the genetic information. So instead, those individuals received the current general dietary recommendations. So for coffee, like I mentioned, it would be to consume up to 4 cups of coffee per day. But if they were in the intervention group and they possessed the risk version of that CYP1A2 gene, they would have been given the more stringent recommendation to limit the coffee intake to no more than 2 cups per day. Now, what's interesting about this type of study design when you're thinking about genetics, that there's different versions of the genotype that the participants can possess. And so we needed to be mindful about that in designing the study. So what we did was actually double the number of participants who would be randomized to the intervention group, because we were cognizant that the prevalence of these genes that confer the increased risk in terms of their relationship with diet and a health outcome, it's about 50%. So half of individuals who were recruited into the study were likely to possess the risk version and the other half wouldn't. So if they didn't possess the risk version, there was really no need to give them the more stringent dietary advice. So we did split the intervention group into 2 sets. And so if these individuals didn't have the risk version of the gene, they were just given the general dietary recommendation. So we doubled the size of our intervention group to be able to account for that. And when we looked back at who we actually recruited and what their genetic backgrounds were, we were actually very close to what we had expected, that about 50% did indeed possess the risk version of the gene. Now, we followed these participants for over the period of a year, and over that time, we did 3 data collection points. At the initial data collection at baseline, we did an assessment of their diets prior to giving them any study material, so they had not received the genetic information or the general dietary recommendation report. So we did an initial baseline assessment of what they were consuming. And then we followed up with them 3 months and 12 months later after we provided them with the dietary reports. And so what we saw at 3 months, there were actually no statistically significant differences in terms of what they were consuming. But we sort of saw this pattern that sodium intake was starting to reduce in the intervention group who had the risk version of the sodium-related gene compared to the control group. And at 12 months, that actually did reach statistical significance. So it took some time, but it seemed that over the course of time, the participants did in fact adopt a more healthful dietary pattern. And it is interesting to note that everyone in the study was over consuming sodium at baseline. Sodium is a nutrient that's really prevalent in our food supply, and it's very common for individuals to consume too much sodium in the diet. So that was true in our study participants as well. Everyone was consuming more than the recommendation for sodium. And we did see this reduction of about 300 milligrams per day, which is considered to be a clinically significant reduction of sodium. There have been some simulation studies done in North America that have estimated that if the population in Canada or the United States reduced their sodium intake by about this amount, about 300 milligrams per day, we would really reduce an astounding number of deaths from cardiovascular related complications. So really modest reductions in nutrition or diet can have quite significant impacts at a population level. So it was really exciting to see that the reduction was at a clinically significant threshold. And what was also interesting to observe was the group who received their genetic information, but was told that they didn't have the version that conferred risk for this sodium-related gene, they weren't given a more stringent recommendation for sodium intake, but they were told to adhere to the general recommendation. And interestingly, they started to reduce their sodium intake as well. It didn't reach statistical significance, but it was certainly trending. And so that was encouraging to see because it suggested that if you were in this situation where you learned about your genetic information, but were told that you didn't have a need to reduce something to a more stringent amount, we didn't see that behavior of maybe feeling protected and thinking that you didn't need to be so mindful. We actually saw that individuals still seemed to be mindful about their sodium intake. So the interpretation we had at the time was that really getting genetic information, regardless of what the action is that's tied to that result, seems to potentially have a positive impact, or at least doesn't have that negative ramification or the unintended consequence of having someone adopt a less healthful behavior. So it was encouraging to see this. We also collected a number of surveys throughout the course of the study, just to understand, because it was the first time that this had been done and no research participants had received nutrition-related genetic information prior to the study. We wanted to understand their perceptions and their preferences for how to obtain this information. And it was quite interesting to see that the intervention group was reporting that this type of dietary advice was more understandable to them. They seemed to understand sort of the role of the nutrition in terms of the health consequence better when it had genetic information included. They found that the DNA-based advice was more useful than generic advice. And there was very little indication of discomfort in learning about this. And so we looked at sort of these anxiety and psychological impacts because there's some literature that was concerned about genetic testing in general, that people may have a hard time understanding, or not understanding, but have a hard time in terms of the mental health or the anxiety related impacts, that perhaps they would feel that this is a bit deterministic and worry about whether or not they would develop a health condition later on because of their genetic results. But we did not see very much discomfort in this process. And potentially it's because it's related to nutrition and it's more actionable. People could modify their nutrition in different ways. So we didn't see much evidence of anxiety or discomfort in learning about nutrition-related genetic information. And what was interesting was there was an overwhelming preference to include healthcare professionals in this process of obtaining this information. And overwhelmingly, it was registered dietitians that were reported to be the most appropriate healthcare professional to be involved in this kind of testing. But only one participant actually shared this information with a healthcare professional. So there seemed to be a bit of a mismatch that the participants had a an interest in a healthcare professional being involved, but we did study them for the period of a year. And out of the 150 that we had, only one of them informed us that they had shared this information with a healthcare professional. So it would be interesting to look over a longer period of time to see if this information gets shared with healthcare professionals at some point. And that's a particular interest that I still have now as a faculty member at McGill, is how are individuals using this type of genetic information in their healthcare management, and whether or not they discuss these results with their healthcare professionals. So I'll just briefly give you some more overview of some previous training. This was a study I was involved with at Harvard Medical School when I was a postdoc. It was a very interesting study. It was one of the largest to look at consumer genetic testing. So I'm not sure, we can maybe during the discussion talk a little bit about whether or not anyone's had experience with 23andMe or other genetic tests that are online like Ancestry.com. But we had one of the largest cohorts of study participants who had actually been consumers of these tests and had paid for them, gotten their information, and we were able to survey them about their experiences. And so in this particular analysis, we wanted to see what were the lifestyle changes, the diet and exercise changes in our sample. We had 1,000 consumers included in the investigation. As you can see, they were fairly high income individuals. 40% or so had incomes of over $100,000. They were middle-aged, and most were what we would consider overweight in terms of their body mass index. And so we had this hypothesis that maybe the actions taken following a consumer genetic test would differ according to someone's perceived health. So there was a question called self-reported health, where it was a 5-point scale ranging from poor to excellent, and the participants rated how they perceived their own health status. And so we separated them into those who reported quite a high degree of self-reported health, very good or excellent, and then those on the lower end of the scale. And so we looked at changes in fruit and vegetable intake and various levels of exercise. And I'll just kind of go over the stratified analysis with the self-reported health, because it was interesting that amongst those who reported lower self-reported health, they actually increased in every measure. So consumed more fruit, more vegetables, and had increased time spent in every level of exercise. So these are fractions, these numbers are means and confidence intervals for the analysis that we performed. And they're small, so it shows that it's not a very dramatic change in their lifestyle, but some modest changes were detected in our sample of consumers. So it's quite interesting. And the fact that it was restricted to those who reported lower perceived health. And actually in the higher perceived health group, we saw a very slight decrease in terms of the amount of light exercise that was performed per week. So it sort of signals that the actions that people take after they undergo this type of genetic testing, it seems to differ depending on how they perceive their health. And so if they perceive themselves as being at a lower state of health, they likely are a bit motivated to improve that. That's possibly part of the reason they even purchase these tests, because they are something that they pay for out of pocket, and depends on which test they took, but some of them are not the cheapest tests. So they certainly would have been motivated, I think, to seek something out that might help them motivate themselves to improve their lifestyles. So it was interesting that in the lower self-perceived health group, we saw more evidence of these lifestyle changes. So this work that I had conducted is a bit different from what others had done. So I kind of sat down and looked at the synthesis of evidence. So these two screenshots of the article titles are both meta-analyses. It's a collection of articles where the data has been pulled together, and an analysis has been performed to look at an overall pooled effect. So there had been two systematic reviews performed about 6 years apart. This is actually the same research group that did both of these reviews. And they pooled all the data on any kind of study that was related to genetic testing and some kind of health behavior. And in both cases, they reported that there was little or no effect of revealing genetic information on health behaviors, But in both of them, they did acknowledge that there may be small effects on diet. It wasn't convincing, but they saw some modest evidence that the genetic information seemed to do something in terms of dietary modifications. And that was a message in both of these systematic reviews. So in the field that I am in nutritional science, and particularly the topics that I'm studying, I find this really fascinating, that there's something about nutrition that people are a little bit more willing to take action on, as opposed to some of the other behaviors that were looked at were exercise or going for health screening tests like mammograms or different kinds of immunization schedules. And so something about diet where people seem more willing to give it a try to make a change. So I'm really interested in continuing on studying this area, but I'm trying to move beyond just looking at single genes. And there's really been a big movement in the field of genetics to also go beyond thinking about single genes and start to look at genes as a collection. And so we know that particularly for diet-related diseases like diabetes or heart disease, these are complex diseases that involve a variety of genetic and also environmental factors. And so I've moved beyond just the single genes to look at what's called a polygenic risk score. And these are a collection of genetic variants that have all been identified as being significantly associated with some kind of health outcome. But one of the limitations around these polygenic risk scores is they're derived from these genome-wide association studies that really are just patterns of statistical relationships between the genetic variants and the outcome of interest. But they have ignored environmental exposures. And so there's a limitation there, that they're valuable in terms of identifying genes that are of interest to a particular disease, but they've neglected the environmental component entirely. So I'd like to spend a little bit of time talking about the importance of the environment in nutrition and weight management in general. In Canada, we have really a high proportion of our population that is overweight and obese. And it is concerning because in the United States, we've seen that the life expectancy has started to decrease in recent years. And the top causes of death in the United States are heart disease and cancer, which we know overweight and obesity are substantial risk factors for both of those diseases. And so part of my concern is that if Canada stays on this trajectory of a high prevalence of overweight and obesity, we may be in a similar situation as the United States in the near future. So it is a priority to think about weight management in our country, but we know that weight loss interventions are very difficult and generally have low success rates. It's very hard for people to stay on a calorie restricted diet, and also finding the time for physical activity can be challenging in our busy lifestyles that we have. And in nutrition, we have this little saying that the best diet is the one that you can stick to. Because really at the end of the day, it's about adherence. It's about maintaining that kind of dietary pattern that will keep somebody's weight within a manageable area. But it's more complex, and this is what I'm trying to reveal in my research program, that this traditional notion of energy in versus energy out, while yes, it's true physiologically, if you consume more energy than you burn, you will gain weight. And if you expend more energy than you consume, you will lose weight. It's very challenging for individuals to make these dietary modifications. So I'd like to do a little bit of audience participation if I can. I like to do this just to kind of get us all thinking about the same phenomenon. So I know we're in the winter term basically now. It's snowing today. It's starting to snow again. But if we think about the summer, it's not that long ago. Maybe if we think about previous summers, because given the pandemic, there were not very many gatherings or barbecues this year. But I'm sure we've all attended a summer barbecue where there's been just a lovely spread of food and a nice variety of things. And you take a few different dishes and you try a few things. And then you realize that you've really eaten a lot and you're very full and you're very satisfied and it was lovely and delicious and you had a great time. But this was just the main course or the various appetizers and entrees that were served. So you've had your fill and you're feeling good, you're feeling full. And then somebody brings this out. And so let's say you're feeling very full, but this looks so good. how many of you would try a piece anyway, despite the fact that you're very full? I would. So I can't see your faces, unfortunately, but I would imagine that many of us are in the same boat, that food is one of those things that's just very appealing, and people can generally make a little bit of room for one more thing, even if they're feeling quite full. So I'm really interested in that behavior. I've put here, this is sort of the basis of ingestive behavior. There's homeostatic or physiological and biochemical regulation of our appetite. But I'm quite interested in these non-homeostatic factors because I find these to be the things that we can intervene on. So cognitive profiles related to impulsivity or learned behaviors, the hedonic factors around taste and how enjoyable food is, and also the social factors. Particularly, I'm interested in retail food environments and the price of food. So I only want to spend about 5 to 10 more minutes because I'd like to take some time for questions, but I'm building this argument of obesity or weight gain as being this neurobehavioral condition and not just this physiological phenomenon of an energy surplus. But our brains, we have this dopaminergic pathway that's very strong, and people are sort of hardwired to respond in the same way when there's been an exposure that's very pleasant. So with food, you consume something, you really like it, your brain's going to remember that. And every time you have that exposure, so if we think about that chocolate cake, there will be an impulse that, oh, I really like that, I should eat that again. And it's very difficult to go against that sort of natural instinct that we have because of the way that our neurobehavioral reward circuit is programmed. And what's really interesting is that if you look at the collection of genes that are involved in obesity, and these are the ones that have been identified by the genome-wide association studies, a lot of them are expressed in the central nervous system, so in the brain. So it adds to this argument about obesity being a condition related to neurobehaviors rather than just metabolism. And compounding sort of our sensitive brain pathways to these food stimuli and food exposures is our modern day retail food environment that is just abundant with food, a lot of palatable foods and energy dense foods. In the upper corner here, I have an image of a food court that you'll see in a mall or various plazas where we know that the nutritional status of these foods is not very good for us. But I'm also building an argument around grocery stores because they tend to be viewed as being a source of healthful food. And while that's true, yes, there's a lot of fresh produce and other healthful items, there's also the ice cream aisles and the candy aisles. And so the grocery store can also present a challenge in terms of being exposed to a number of highly palatable food cues. So the research questions that I've been looking through in my program here at McGill is how do our genes interact with our food environment to influence what we eat and various outcomes related to adiposity. I'm also interested in looking to see if we can identify a genetic basis for the different cognitive and hedonic aspects of eating behavior. And once I've gone through those projects, I would like to start to design something like what I presented at the start of this about an intervention study, a randomized controlled trial where we disclose genetic information, but instead of just disclosing it in terms of nutrition relationships, looking really at eating behaviors and what people may be predisposed to in terms of food preferences and impulsivity around highly palatable foods to see if those types of interventions may be more successful at modifying dietary behavior. So I've got some preliminary results. I've worked very extensively with a Quebec biobank. It's called CARTAGENE. You may have heard of it, but it's a representative sample of Quebecers who are middle-aged, and a nice subset of them have had their DNA analyzed. And so I'm able to look at these relationships with the genetics. And because in Quebec, I've also got access to data on the retail food environment, I can link those two data sets, the CARTAGENE cohort with the retail environment, and they overlap nicely in terms of time and geography of where the participants live and where the food retailers are. So it's a really nice creative way to look at these gene environment interaction questions. These are the main areas of Quebec that have been represented in the linked data of the food environment and the CARTAGENE biobank together. In the food environment, we've got a lot of information on what's in the retail food environment, and a lot of the stores that are included in this database are grocery stores throughout Quebec. And we've got some nice information on the variety of products that are included in the stores, their prices and frequency of sales of those products, and also their in-store advertising activities. So which types of foods tend to be on display in the grocery store. So if you think about going shopping and you're looking at the aisles, there's often a number of products that are right at the start of the aisle and they're on sale, and there's a promotion of them. So we've got the data on what kinds of food products tend to be displayed for promotional purposes. So this is a statistical analysis that we conducted. I'll just go straight to the results. We looked at, in terms of diet, one of the variables of interest was diet quality. I like this metric because it combines, as you can see, a number of components of the diet, and it looks at foods that are considered healthful as well as those that are not so healthful. And so you get that combined score that doesn't necessarily penalize, but your score will change according to how much of those unhealthy items are consumed by the participants. So it's a nice way of sort of capturing a more holistic measure of diet. So in one analysis that we did with a dopamine gene, the DRD2 gene receptor, that's been associated with various types of eating behaviors, we saw that individuals who carried a particular mutation in DRD2, if they were in an environment that had a high degree of promotion of ice cream products, they actually had a statistically significantly lower diet quality score, which means a less healthful diet, than individuals who had the same genetic variant but were exposed to a lower proportion of those ice cream displays. So starting to see some evidence that our genetic background and our environmental exposures may work together to influence some of the dietary outcomes. And one of my graduate students continued this work and looked at this polygenic risk score of obesity. And she was able to show that diet is important for obesity risk offset. And so this is showing the solid line is individuals who had a lower polygenic risk score for obesity, and the dashed line is those with higher. And so if you had a high polygenic risk score, but a healthful diet quality, the healthy eating index is on the x-axis, eventually, if the diet quality was quite good, the average body fat percentage, waist circumference, body mass index, it was actually lower than individuals who had the low polygenic risk score. So it suggests that those at high genetic susceptibility for obesity may be quite responsive to a healthful diet in a positive direction. What was really interesting was we saw that not only diet quality seemed to offset that genetic risk of obesity, but food prices in the environment did as well. We created a ratio of food discounts measure. And so this considered healthful or unhealthful over the healthful items in a grocery store. And we were able to see that when the healthful items were on sale more often than the unhealthful items, there was this attenuation of body fat percentage, waist circumference, and body mass index amongst those who had the highest polygenic risk score for obesity. So it's not just diet quality that seems to offset the genetic risk amongst those with high genetic susceptibility, but also the prices of foods. So if more healthful foods were more often on sale, we observed that the outcomes with adiposity measures were better amongst those with the higher polygenic risk score. So it makes sense because we do have to buy the foods that we eat. And so if the healthier foods are more accessible and more affordable, our data is suggesting that that could have a positive effect on obesity outcomes. So I'd like to take some time for questions. I know we've only got about 15 minutes. My last section was going to be about taste, but maybe Ingrid, I'll pause, and during the question period, I might be able to get back into this, but I just want to be mindful of the time.
You're right. Thank you. Thank you. Well, let's all open our cameras and our microphones, and let's show our appreciation for a lunch hour talk. That's so fitting. I'm sipping tea, but oh my goodness, just seeing that grocery aisle. And that's actually my first question. But first, proper thank you. First of all, thank you for your accessibility. The closed captioning worked really well. There were some funny ones. You did send your slides in advance. That was also recommended by the accessibility people at McGill for a lot of these presentations. And thank goodness you did that, because a lot of your data, of course, and those informative slides that, they go by so quickly, if they're now available. Fergus is recording, has pressed the record button, but everybody got the slides, everybody who registered, so they can go back to that and read or re-examine or understand more, and at another time. Dr. Nielsen, you'll be receiving an official gift from the Red Path Museum. This is a book called Treasures from the Crust. It's about our mineral gallery guide, and that will be in the mail for you in thanks. So yes, we have about 12 minutes for questions. I just have to tell you about my, what's it called? You called it your ingestion tendencies.
Ingestive behaviors, yes.
Behavior. It's so changed as I get older, and it's really moved into salty, super spicy, or something that gives a really quick fix. Whether that's, that first glass of wine at the end of the week, Friday night, it's great. That first sip is just amazingly powerful. And I don't even need to, it's not like I need to drink the whole bottle or anything. Is that, you haven't addressed age, I think, in all your studies. I don't know if you're dividing men and women, or is it a particular age group?
Yes.
Is that something I can be aware of?
Yeah, it's a very good point, because what I'm increasingly learning from engaging more with this literature and conducting my own work is how much of our eating behaviors and our food preferences are linked to our memories and our experiences with food. So as one ages, you've got more experiences, more memories. So there's a lot of sort of cognitive psychology going on. And I find that really fascinating. And it makes sense in terms of how people have these emotional relationships with food. But on top of that, there is an aspect of sensory perception that also evolves with age. And so when you're talking about salt being more intriguing to you now, and spicy things, that could be because of some age-related declines in other types of sensory perception that are happening. And so there could be sort of a more intense stimulus that's needed to reach this kind of gratifying threshold in terms of the sensory experience. And what I was going to say was that taste and other kinds of sensory signals, they're really experienced in the brain. We think about it in the mouth, and yes, that's where the chemical sort of exposure is happening, and the taste receptors are collecting that information, but it all gets communicated and processed through our brains. And one of my aspects with taste is going to look at the overlap between the sensory perception, because a lot of this is happening really throughout the brain. Particularly, I'm interested in taste, which is processed in the gustatory cortex. But this particular region of the brain also has a lot of our appetite centers, where various hormones are being processed in terms of signaling for hunger or satiety. So there's this overlap between sensory processing and appetite signaling that's really been underexplored in the literature. And so that's another area that I'll be delving into further in my research. I'm really excited about that. But yes, age can play this sort of dual role in terms of how our sensory perception evolves over the course of time, and also how our experiences and memories around eating accumulate over time.
Thanks. We have, I think, a few questions in the chat. Fergus, have you seen anything come through?
Yes, Ingrid, I have. We have a question from Liette. Professor Nielsen, are you able to see that in the chat, or I can read it out for
you? Maybe you can read it out, because when I'm in full screen mode, I don't see the chat.
So Liette asks, my husband and I had several types of DNA tests done, and I wonder if any can be used for nutrigenic evaluation. And if yes, where can we send the info for evaluation?
It's a very good question. I'm not sure exactly what kind of genetic test you had, but what I can say is if your report has provided you with the raw data, there are these third-party sources now. There's websites. One of them is called Prometheus. You can email me if you want me to send you the link. But for genetic tests that have provided the consumer with the genetic information, sort of the raw data file, you can upload your report into this third-party link, and it will go through all of the genetic variants and produce to you this kind of updated report. And it sort of mines the scientific databases. And there's a number of genetic repositories that exist where scientists are trying to compile what's known about various genetic mutations. And so it sort of mines that data and identifies the gene variant and says, okay, now we know it's associated with this trait or this trait. So you can find the nutrition information through these third-party providers. And I believe they used to be free. They may now have this modest $50 fee or something like that, but there are ways of trying to kind of extract more information from your genetic report. So you can email me and I can send you the link.
Thanks. Ingrid, we did receive some other questions in advance. These are from Adina. I might put them in the chat so everyone can see them, but I'll read them out as well. Adina asks, if we don't test to find out which are our problem genes, what would be the healthiest diet for everyone to avoid health problems like diabetes, heart conditions, Alzheimer's? What would a daily menu look like?
It's a very good point. I think that a healthy diet can look a few different ways. I think in general, nutritional scientists all agree that a lot of fruits and vegetables is really a way to go. We want to have high amount of fruits and vegetables, less refined grain products, so more whole grain products, and lean sources of protein, whether it's lean animal protein or plant-based proteins, which the plant-based proteins tend to be lean by default. So a Mediterranean type dietary pattern that's high in the fruits and vegetables, high in the whole grains, kind of modest in terms of animal protein and plant protein. That's a very healthful dietary pattern in general. And that's one of the issues with this field of nutrigenomics. It's that question of equity and access. It seems very unrealistic to expect that everyone in the population would be able to access this type of information right now. It's really on an out-of-pocket basis, and who would like to receive it, who can afford to receive it. So there is the equity consideration. And where I see nutrigenomics playing a role in terms of public health and population recommendations is we can fine-tune those recommendations. I think caffeine is an excellent example. If we have this evidence that demonstrates that 400 milligrams of caffeine per day can be too much for some people and predispose them to various risks, why don't we make the general recommendation 200 milligrams per day for everyone? So I see it as a way of updating the general recommendations so we can cover those who are most vulnerable in the population according to what we know about the genetic relationships. So I'm not advocating for this to become part of public health in terms of everybody getting their genetic information, but I think we can use the evidence to update our public health recommendations.
Okay. So if we have time for a couple more questions, again from Adina. If somebody has family with diabetes, should they eat a low carbohydrate diet before getting to the point of becoming pre-diabetic or diabetic to avoid it?
Yeah.
How many carbohydrates per day would be okay to eat to possibly avoid diabetes in this case?
I saw this question. It's a very good question because the issue about prevention and family history, it's really important. And I'm sorry that I don't have a very concrete answer for you because I think a lot of those questions are really being carefully studied now. In fact, we have a faculty member in my department, Professor Anne-Sophie Brazo, who is an expert in diabetes, and she's particularly interested in carbohydrate intake and carbohydrate quality. So she would be a good person to touch base with if you want some answers to those questions. But I know that her work is in progress to delve into some of those questions because from a treatment perspective or a therapeutic perspective, there does appear to be benefit to carbohydrate restriction. But to someone who doesn't yet have diabetes, we're not so sure that it's a beneficial thing. We're not 100% sure that it's safe. So there are general recommendations. If you look up the dietary reference intakes, these are the standard public health recommendations. They're called the DRIs, dietary reference intakes. You can look at them online. They were developed in consultation between American and Canadian researchers, so they pertain to all of North Americans. And there are guidelines in terms of consumption for all the macronutrients. So the carbohydrate recommendation right now is that they make up about half of our diet, 50%. It goes from kind of 45 to 65%. So on average, you can say about half of what you consume could be from carbohydrates. But I would say that carbohydrate quality is also very important. If you look up the work from Professor David Jenkins at the University of Toronto, he's done a lot of work on carbohydrate quality and the glycemic index. That does make a difference in terms of the glycemic response to food. So how our blood glucose levels respond to what we eat. There's a large impact of the carbohydrate quality. So more complex carbohydrates are better, less refined carbohydrates. But in terms of restricting them, if someone does not yet have diabetes, it's unknown whether there's a benefit to that. But it's a good question about family history as well. That's something that needs to be explored if for those who may have a higher risk due to family history. So again, I'm sorry I don't have a concrete answer, but I wouldn't advocate for carbohydrate restriction if someone does not have diabetes. And I think that there can be a healthful diet with a good quality type of carbohydrate at about a 50% level of your food intake.
Okay, thanks. And thanks, Dina, for the question. A couple more have come in. One from Liette about green tea. Is green tea healthful, and is there a maximum amount not to exceed?
It's a good question. So I think all teas are quite healthful, actually. They tend to be lower in caffeine than coffee. Green tea in particular is not very high in caffeine. So if you're looking for an alternative to coffee, green tea can be very healthful. What's really interesting about green tea is the phytochemicals and the flavonoids that it contains. And there's a lot of interest in understanding the role that those kinds of food compounds may have in our health. I wouldn't say there's a lot of really convincing evidence about any major real benefits. That's the thing about nutrition, is it tends to be these small cumulative effects. So yes, consuming green tea may have some modest benefits in terms of the flavonoids and the antioxidants that are present. But as a kind of magic bullet, so to speak, I don't think that is there. But if you're looking to replace green tea versus coffee, I think there could be a benefit to that if somebody has a need to be mindful of their caffeine consumption. But it's really, and I think the benefits of nutrition are more holistic. You have to look at really everything that everyone is consuming. It's actually really interesting because right now in the pandemic, I've been studying the food impacts of the pandemic in Quebec and also other provinces in Canada. And we're commonly seeing our survey respondents report that I'm eating better, I'm cooking my meals at home more, I have more fruits and vegetables. But alongside with that, they also talk about baking more and baking muffins, baking cakes. So it's sort of, we don't know the net effect yet. We're trying to look at that to understand. People think they're eating better, but if there's a lot of this baking and sort of dessert consumption, we don't know if indeed the net effect is a health benefit. So that's the thing about nutrition. You have to think holistically in terms of the full picture.
This is a very interesting talk, and it's prompting a lot more questions coming in through the chat. Ingrid, I defer to you as to how we're going for time.
So I think the best we can say is just thank everyone who is involved in this as participants, as speakers, Fergus as our tech. Nicole, thank you for that introduction. And how about if we could direct those questions directly to Daiva, to Dr. Nielsen, Fergus, if that can be sent by email, and she would promise to answer them for us. Does that make sense?
Yeah, that's absolutely fine by me.
Give her a little bit of something to do now that she's finished teaching classes. Yeah. She told me that she has a bit of a gap now. We also have a gap. We won't see each other again until January 20th, 2021 next year. That that Wednesday Stars lunch hour speaker is Heather McShane. She is with MISSI, that's the best acronym at McGill, the McGill Institute for Sustainable Science Initiatives. Heather has her PhD in soil science, and actually that's the title of her talk is what was her journey from soil science or soil chemistry to sustainability. There it is. That's wonderful. You get a sense of the stars and what's in store for us in a few weeks. Actually, it's not far away.